All right, online, can you see my screen?
Yes.
Awesome, thank you. Can you hear me okay also?
Yeah, I can.
Okay, great, thank you. All right, so we're gonna get into it. We have 2 more lectures today to cover. I'm short, so there we go. 2 more lectures today to finish up our physiology kind of anatomy discussion. This lecture in particular also does some coverage for immunology. So there's going to be a little bit of crossover here because the immunologic system is an organ system, but it also is a separate part of your exam, specifically for the pathology, disease and dysfunction section. So this lecture, our purpose is to kind of set you up for success for next week when we go through cytokines. So next week, we're going to focus on cytokines in the immune system, and then a couple other kind of funky mechanisms. So this here is going to talk through all of our different cells from the top, from our HSC cells, our hematopoietic stem cells, all the way down to our blood cells, our lymphocytes, our granulocytes, all those good things. And then our last hour, we'll spend focusing on repro, some high yield concepts. I tried to sum it up as much as I could. All right. There was a great question during the break about how much of this is clinically relevant? How much of this will I use in my clinical practice after taking this test? Probably not a lot, okay? A lot of this is stuff you'll look up if you don't know. A lot of this is good foundational knowledge, but it's not necessarily going to be stuff that you're like talking about on your day-to-day with your patients. NPLEX 2 feels that way. So your second board exam will feel very integrative, feel very much like, oh, this is really helpful for my future as a doctor. This one, not so much. So this one is really that foundational knowledge. I don't like calling it a gatekeeping exam because it's not its purpose, but it's really making sure you were here for the first 2 years and you have that baseline knowledge to help propel you into your next phase of your medical program. But clinically, I don't think I've mentioned enzymes often in clinical practice. Okay, so the dance of immunity. So our cells. Our goal is to successfully vanquish pathogens. To do that, we have to complete a wide variety of activities. Our innate immune cells come first. Our antigen-presenting cells then will communicate that an infection is present to our lymphoid cells, which is part of our adaptive immune system. They'll coordinate the adaptive immune response. That gives us some memory. So next time when we see the thing again, we can respond faster and greater. So that's really a gist of what we're getting into here today. The specialized anatomical structures and microenvironments help organize these processes. And I'm going to try to help you lump them into categories today to help with memory. So we have 2 systems that kind of are the organs that are involved in this process of our immune system and our blood. Our primary lymphoid organs, we could say these are the primary organs of the immune system, is bone marrow and thymus, and it's essentially where immune cells are developing. Bone marrow are B cells and really all other cells, and then thymus, just T cells, T lymphocytes. Our secondary lymphoid organs are the organs where these cell types are going to differentiate into their memory cells or their effector cells that are actually going to do the effects on the body. This is your spleen, your lymph nodes, and any specialized organs in the gut or mucosal tissues. So Peyer's patches, right? These specialized tissues or structures found within the body. You'll also see that these are the sites where your antigen-specific lymphocytes will find their antigen counterpart. So this is where they become specialized, is in these secondary lymphoid organs. The systems are connected by blood and lymphatic vessels. It's important to remember that blood is a highway as much as lymph, and they interchange, and oftentimes where they communicate is the spleen and the lymph node. That's where blood and lymph systems are going to be able to communicate, share information, and share cell types. Of course, cells can leave blood vessels via diapedesis, D-I-A-P-E-D-E-S-I-S, I think. But essentially, it just means cell escaping the blood endothelium into the tissue around. So this crazy thing is what we're going to be looking at. This is the differentiation of immune cells, so hematopoiesis. The process of how we get from a stem cell to our myeloid progenitor or lymphoid progenitor and to all of its products. So a great place to start when it comes to learning these different cell types is to be able to separate them between myeloid and lymphoid. Lymphoid are your lymphatic cells, your T cells, your B cells. That's primarily on the lymphoid side. There's a few other specialized ones that you can see, like a dendritic cell is on both sides. and an innate lymphoid cell, which has lymphoid in its name. So that can help you remember it. Our myeloid side has everything else. All of your granulocytes, including your macrophages, your monocytes, your eosinophils, basophils, etc., your mast cells, your megakaryocytes, which become platelets, and your blood cells, your erythrocytes. So myeloid progenitor has much more biodiversity than your lymphoid cells, but your lymphoid cells can become much more specialized, right? Those are ones that can become specific for antigens like gluten or gliadin, versus your myeloid is going to have probably one general job it does, regardless of what it encounters. Okay. All mature red blood cells have the same ancestor. That's why I just mentioned here. So from this picture. Stem cells, HCS cells have, or HSC, hematopoietic stem cells. Is that HSC or HCS? HSC. I always want to say HCS because that's the school I coached at. So HSC, hematopoietic stem cells, have two different abilities. They can regenerate or self-renew, and they can differentiate into diverse cell types. So they could become their germ cell again, where they become their own cell that can self-renew to continue the process, or it can actually diversify. Embryonic stem cells, we can see there's two different forms. So embryonic stem cells are pluripotent. That means they can generate almost every type of cell in an organism, right? So how we're able to start with a zygote and turn into a whole, from a fetus to an actual baby to an adult. It all comes from a pluripotent stem cell. Adult stem cells are multipotent, the capacity to give rise to diverse cell types that are particularly focused on a tissue. So in adults, we have certain organs that can regenerate, like the liver being one of them. In that liver, there's multipotent stem cells that can regenerate and regrow tissue that gets damaged or destroyed, okay? So you can see the difference between the types. They love to throw in pluripotent, multipotent, throw these words in questions to try to pull you or sway your answer. So knowing these definitions can be helpful. Our hematopoietic stem cells differentiate into all of our red and white blood cells. It was the first tissue-specific stem cell identified, and we can just remember it as the source of our erythroid and our leukocytes. So we're going to look at how that happens. So our multipotent progenitor choices. We have our myeloid or common progenitor cell, that becomes all of these things that I already mentioned. It's really going to be known for producing our innate immune system. The immune system that responds first and responds the same way every time. Okay. Our common lymphoid progenitor cell creates our lymphocytes or B and T cells, as well as the innate lymphoid cells, which these have innate immune properties. So while a lot of the things on this side are part of the adaptive immune response, we can see here that both of these innate lymphoid cells and dendritic cells are going to have innate immune responsibilities as well, not just adaptive. Okay. So the myeloid lineage is the first responders. Our red blood cells, our granulocytes, our monocytes, our macrophages. They're going to respond rapidly, and they're going to communicate a pathogen is present. So our granulocytes. So if we go from myeloid as a category, we're now going to subcategorize into a few different buckets. One bucket is going to be granulocytes. There are 4 granulocytes you need to know: neutrophils, eosinophils, basophils, and mast cells. The common kind of denominator between all of these is that they all are going to have, for the most part, multi-lobed nuclei, except for mast cells, they don't really, but the majority of them have multi-lobed nuclei, and they're going to have some type of granule that they can release. These are first responders to immune responses. So focusing on our neutrophils, these are the majority of leukocytes in the blood circulating or in adults. So this is the major one. If they ask you what's the most common or the most numerous leukocyte or granulocyte, neutrophil would be your best answer. Okay. They're released into the peripheral blood. They circulate for a while until moving into the tissue. Once they're in the tissue, they'll live only a few days. And their big job is generating inflammatory molecules called chemokines that promote development of more neutrophils in the bone marrow, so they can upregulate their growth and development of neutrophils or differentiation of neutrophils. They also do something called the neutrophil swarm, where if one neutrophil finds a source of infection, it can essentially recruit with chemokines other neutrophils to that site And then it's almost like pheromones. They're drawn to those chemokines, and then they can source or mount an attack against whatever antigen is present. They can phagocytose bacteria and secrete antimicrobial proteins and tissue remodeling proteins. So they can not only phagocytose, they can directly secrete and destroy via proteins that are antimicrobial, and then they can also remodel tissue to start that repair or inflammatory response or process. If you have lots of neutrophils that are circulating, that can be a representation of leukocytosis or infection. You can see they have the multilobed granules present in their nuclei, or multilobed nucleus. They have granules present, that's these things, and phagosomes. All these little purple dots are their granules, and then these are the nuclei. Very pretty. Eosinophils, they are going to have granules that stain pink. So that's a big differentiation between a neutrophil and an eosinophil. We can see that here in this picture. These granules are much more pink than our neutrophil image. They are going to coordinate defense against extracellular stuff like parasites, helminths, worms that are in the body. They cluster around them and can damage them by releasing their granules. So essentially, eosinophils are going to surround a parasite, surround a worm, surround a helminth, and they're going to start releasing their granules to essentially digest that worm locally in the body and destroy it. They are motile. They can migrate from blood to tissue. They're located highly concentrated in the small intestine, which makes sense that they respond to parasites. They can also, though, contribute to allergies and asthma symptoms because they pick up on antigens that are outside of cells. If we eat something, if we consume something, if we breathe something, if we locally have something that contacts our skin that we are allergic to, eosinophils, it's not going to be inside of a cell. It's going to be floating around in our interstitium or our blood or in our respiratory system or somewhere in our body that's not engulfed inside of a cell that we have. And so our eosinophils can pick up on that. They can identify that as not self and mount an attack or mount an initial response. So that's why you could have eosinophilia related to a parasitic infection or eosinophilia related to an allergic attack or allergic response, atopy, asthma, eczema, allergic rhinitis. Basophils. I mentioned our basophils. That was my kicker to keep you here, right? They are non-phagocytic granulocytes. They do not phagocytose. Their basophilic granules stain blue, so that's why they're so dark. I think of basophil blue granules. They are very rare. They respond also to parasites or helminths, and they can bind circulating antibody and antigen complexes first, and then they will release their contents of the granules. So basophils are interesting in that they can actually respond to an antibody antigen complex. So that is a part of the adaptive immune system too, right? Anything with antibody is adaptive because it means our immune systems had to produce that or see that before. So basophils can respond innately and they can respond adaptively in that sense, which makes them unique. Another unique component of basophils is they are a histamine producing granulocyte. So they can release histamine, which is in their granules, which increases permeability of blood vessels. It increases smooth muscle activity, allowing for vasodilation, essentially more blood flow to the area and allowing easier diapedesis, stuff to be able to leave the blood and go into the local tissue. That's going to allow for more immune cells to come to that site quicker. You'll also see a release of cytokines to recruit other types of cells. They can cause allergy symptoms. This is the most rare granulocyte. So I see asked often, what's the most common granulocyte? Neutrophils. The most rare granulocyte, basophils. Right, and then mast cells. These also can combat parasitic worms. They are contributing to allergies as well. We often think of, we talk about things that can stabilize a mast cell, right? Stabilization of a mast cell is just preventing degranulation of a mast cell, avoiding the granules from the mast cells to be released. That's what we're talking about when we say, we're going to give you something to stabilize your mast cells today. That's what we're hoping to do. Mast cells are released from bone marrow into the blood in an undifferentiated state. Then they mature once they leave home. So this is different than the other types of granulocytes that mature first in the bone marrow before leaving home. Mast cells get to leave home a little bit early on, and they kind of grow up as they travel through the body, through the blood. And once they get into the tissue. We find them often in skin, connective tissue, mucosal epithelial cells of the respiratory, the GU, and digestive tracts. And they have lots more granules that have a lot larger content. You see these granules are much larger than even basophils. So while basophils can release histamine, mast cells are much better at it than basophils, and they're much more commonly found in our body and in our blood. So our majority histamine producing thing is going to probably be a mast cell if we had to choose one cell. Basophils produce it, but there's just not that many of them. So mast cells, if we have a really big histamine response, it's mast cells that we want to target. Okay. So if you want, if you need something to kind of differentiate these three, you can see neutrophils, eosinophils, and basophils and mast cells we combined here together because they do both have very similar action. These are the molecules that are present in their granules, what those molecules specifically could be called, and then what specifically is their function, right? How much of this should you memorize? Know the things on the previous slide first. So that's your baseline understanding. Whatever else in details you can get to, great, but you have 2 weeks, right? Less than 2 weeks. So keep that in mind when you're dealing with the minutia of things, right? At this point, maybe not the best time to remember every single one of these acronyms. We will do a cytokine like deep dive next week that will help you kind of differentiate the important ones, but I wouldn't memorize the whole table here. I would generally know like what these functions are. That can be helpful to understand kind of what they're doing in general, but recognize neutrophils primarily are going to focus on things intracellularly for the most part. So that's why bacteria, viruses. Eosinophils extracellularly, parasites, helminths, worms. And then basophil mast cells are really your big histamine producers, your allergic responders, but they are more rare than the other two. Okay. All right. Any questions about granulocytes? That was our first category of our myeloid lineage. Okay. Our next category is our antigen presenting cells. These are our myeloid lineage cells that present antigens. So they're going to be really, their big job is to alert the immune system that something is wrong, that something that's not self is present. This will include also cells that do phagocytosis, like our monocytes, our macrophages, and dendritic cells. And we call this professional antigen presenting cell function, so PAPC, or you could see it as just APC. So immunologists will state both. So our professional antigen presenting cells, there's 3 major activities that occur when these cell types encounter an antigen or encounter a pathogen. They're going to secrete proteins, chemokines that attract and activate other immune cells. They're going to phagocytose, and they're going to break down these pathogens into smaller peptides and then present these peptide antigens on their surface via MHC, major histocompatibility complex class 2. That's important for us to remember. The class 2 presentation is a specific cell or skill. Only MHC class 2 molecules can communicate to T helper cells. So our professional antigen presenting cells, or APCs, are the ones that can alert a T helper cell that something is wrong. And that's really important because our T helper cells have a big role in our adaptive immune system. They also will then upregulate co-stimulatory molecules, cytokines, to activate helper T cells, to send cells to the site of infection, and essentially initiate the adaptive immune response. So where you could say granulocytes are the initial defenders, right? Granulocytes are actually defending locally. You could say that the professional APCs are like your commanders in chief. They're going to communicate what's happening on ground to your specialty team back at home, back at base. Okay. Monocytes are the first one we'll look at. 2 to 12% of white blood cells, so again, a smaller amount. They can migrate into tissues, and then they can differentiate from there into its phagocytic cell type. There's an inflammatory and a patrolling type. The inflammatory type is going to enter in quickly in response to infection, and once it's there, it's going to secrete a lot of cytokines, cause a lot of messages to be sent out. It's part of our initial inflammatory response. The Rubar, Calar, Dolar, whatever the other ones are. The patrolling type is going to kind of crawl around slowly along blood vessels, and it'll monitor blood vessel repair and also any blood vessel damage or sites potentially of attack. This is the type potentially that can be involved. Oh, my little clicker is dying. That could be involved in atherosclerosis, right? If we have damage or if we have a fatty streak present and we have our patrolling type monocytes can identify that and then send a signal saying, hey, we need to fix this, we need to clean this up. And initially that's okay. And then over time that can cause more and more plaque buildup locally. Our macrophages are what we know them to be. These were initially monocytes that then have migrated now into tissues in response to infection. They are the best at phagocytosis. They're part of our innate immune system. Their main job is to clear pathogens and present them to T cells. And there are many specialized types that have certain names, and we'll look at that here. We've already seen a few, right? Microglia, our neuroglia in the brain. We've seen, you may remember a Kupffer cell in the liver, Langerhans cells in the skin. So probably the three funky named ones that I would remember the most. But these are all macrophages. They're just specific to that tissue. They still all will have the job of phagocytosis and professional antigen presenting cell abilities, but they may also have some other abilities specifically to the tissue that they are located in. So of these, I would only really focus on brain, liver, skin, those three, because they have different names. Once we have presented part of the job for a macrophage is to present these molecules to T cells, but to do so, they have to phagocytose. And a lot of our bacteria specifically have virulence factors that protect them from being phagocytosed or protect them from being essentially engulfed. And one of those virulence factors is a capsule. Any bacteria that are encapsulated or have capsules essentially cannot be phagocytosed. They have to first be opsonized, and that is essentially they have to be lined with antibodies before macrophages can recognize and be able to actually phagocytose them, digest them, and present them. So why encapsulated bacteria cause worse infection typically, and the infection can last for longer because it can take a while for the body to actually recognize an infection's present. So opsonin is another word for an antibody. So that's why you have opsonization, essentially covering a foreign bacteria or a foreign substance with antibodies. Once you've done that, macrophages then can identify, phagocytose, and present that to T helper cells. Okay. The next professional antigen presenting cell is a dendritic cell. Dendritic cell is different from a dendrite on a neuron, right? We had the dendrites, the projections from the neurons that kind of like are feeling the environment around a neuron. Dendritic cell is different than that. It's actually, but it kind of looks the same, right? It has these like kind of spiny projections, and it's helping us initiate our immune response. These extensions actually look like nerve cell dendrites, and they help to increase surface area to browse lymphocytes, to browse cells, to see what they can find, see what the body has been exposed to lately. You'll see them in both myeloid and lymphoid lineages. So you know they must be important because the body has two ways to create them. So it doesn't want to, if one part goes wrong, it still has a backup plan. So this, again, I think of this as your 911 director, right? You call your 911 operator and say, this is what's going on. The 911 operator then decides who to send to that site, who to send to that location. That's your dendritic cell's job. If you're having a hard time visualizing these cells, I encourage you to look up cells at work. It's like a probably not 1900s, probably like 20 or 2015, 18 like show. It's anime, but it was created by med students, and it goes through, you're a red blood cell, that's the main character, and she encounters all these different pathogens. You meet all the different immune cells, but it's like at the level of a medical student knowledge. So it is helpful if you're looking for like a fun way to learn the immune system. Cells at Work. It used to be on Netflix. They took it down, but you can still access it. Just Google Cells at Work and you'll find it. But their main job is to capture antigens and present them. That's what their main goal is. And they can communicate with lots of different cell types. Okay. Their presenting function, immature dendritic cells can take in an antigen in multiple ways. They can do phagocytosis. They can do endocytosis by binding via a receptor, or they can take it in by pinocytosis. All of these essentially are engulfing, putting something into a vesicle within the cell. I do not expect the NPLEX to have you differentiate between these three types, okay? So again, they're engulfing something and taking it internally. Once they have an antigen that they have engulfed and presented, they will change essentially their phenotype. They'll change how they express themselves to be able to specifically present the antigen to T cells. That's the big person they're communicating to. Once they are becoming a professional presenting cell, they cannot phagocytose anymore. So once they've presented something, they've lost the ability to phagocytose. They essentially differentiate into just an APC. When they are mature, meaning once they have actually identified a specific antigen, they will, or they have a specific antigen that they bind with, they will migrate through lymphoid organs like the thymus and lymph nodes and spleen and bone marrow. They'll specifically hang out in, well, really all of them. So I'll say they hang out in all of them, and they present antigens to our T cells that circulate throughout the body. Follicular dendritic cells are different, so keep that in mind. I'm talking about dendritic cells here, but follicular dendritic cells, FDCs, they arise from a different lineage. They are different. They are functionally distinct. They're in follicles within the lymph node that B cells are present, and they're regulating the activation of B cells. So if you hear follicular dendritic cell or follicular dendrite, that's different from this antigen presenting dendritic cell I'm talking about. Your typical APC dendritic cell communicates to T cells. Your follicular dendritic cell communicates to B cells. Okay. All right. Now the blood. Our erythroid cells, erythrocytes and red blood cells. These cells have high concentrations of hemoglobin. Their main job is to deliver oxygen to cells or tissues. They can release signals like cytokines that induce innate immune responses. They are anuclear, which means they're going to not divide, right? They're anuclear. And they generate nitric oxide, which can also damage microbes and vasodilate, helping them travel faster and further. They'll have surface receptors for antibodies, and they can bind antibody complexes. If they do, they'll then be cleared by macrophages. So we're going to go through our formation of red blood cells. This is tested on relatively often, so I think this is a high yield to at least go through this process. So we start here with our HSC cell. We're then going to become a myeloid stem cell. So this would be step one. This would be step two. Our myeloid stem cell will then become a pro-erythroblast. Our pro-erythroblast is going to divide a bunch of times, and then we get a basophilic erythroblast. I don't know if I have enough here. We'll see. Our basophilic erythroblast will have lots of ribosomes present still. Okay, so it's still going to synthesize lots of ribosomes. We're still we're still evolving. That's one of the ways we can tell the difference. Our polychromic, here, I'm going to change my thing here. Our basophilic erythroblast is going to become a polychromic erythroblast. This is where we start to see synthesis of hemoglobin. Our polychromic erythroblast is then going to become an orthochromic erythroblast. This now we see a small number of ribosomes. And then, oh, I did have enough steps. Okay, never mind, sorry. And then our orthochromic erythroblast is going to become a reticulocyte, which still has a small number of ribosomes. And then our mature erythrocyte is going to be our final stage where we are anuclear and we have lost all of our ribosomes present, resembling much like a mature red blood cell. Here, 4, 5, 6. There we go. They do mash. What ones would I want you to know? HSC, myeloid, proerythroblast, definitely those three. Reticulocyte and erythrocyte. You know those 5? Great. So the first 3, the last 2. If you can remember the ones in between there, fabulous. But if you remember the first 3 and last 2, it's going to get you through a lot of questions. When do ribosomes go away? That's once you're erythrocyte. So that is potentially a question you'll see is like, when are you in your final form with no organelles, no nuclei? That's erythrocyte form. There's some questions about color of cells, because you can see it switches color from 4 to 5. It switches color and turns more red once hemoglobin is started to be synthesized within. So if there was a question on color change, I doubt they would ask that. but that's why you can see a color change here in that image between 4 and 5. It's because of the production of hemoglobin. Erythropoietin EPO is your hormone that helps to regulate this process. So recognizing if we had a stimuli of low oxygen or hypoxia, low oxygen delivery, due to decreased red blood cell count or decreased hemoglobin or decreased availability of oxygen, the kidney and a little bit the liver would release EPO. EPO essentially goes to your bone marrow, stimulates red bone marrow to produce more cells, specifically differentiate them into erythrocytes. So you'll have enhanced erythropoiesis, which increases your RBC count. And then you're going to see that your oxygen carrying ability of blood will increase because you have more blood cells present. Testosterone can also increase EPO, which is why you can see elevated red blood cell count in people who are taking testosterone exogenously or have high levels of testosterone. So some nutrients that are necessary, obviously iron and vitamin B12 and folic acid are the big ones. Iron you can get from diet. 65% is found in the form of hemoglobin. The rest is in the liver, spleen, and bone marrow. Free iron ions can be toxic, so it has to be bound. Stored in cells, we call iron ferritin and hemosiderin. Transported in blood and bound to protein, we call it transferrin. So when we're measuring these things, we can often measure potentially all three. We often measure ferritin first. We can also measure transferrin and other things and free iron as well, but it's toxic. So you wouldn't want that around too much on its own. B12 and folic acid you need for DNA synthesis for any rapidly dividing cell, right? And so if you're undergoing red blood cell creation, erythropoiesis, that's a rapidly dividing cell. You need to then have vitamin B12 and folic acid to have adequate erythropoiesis, adequate red blood cell development. Hence why we can have anemias, right? Our megaloblastic and microcytic anemias. Our life cycle, I have just this picture here. It's important to remember that RBCs live for about 100 to 120 days. So that's their life story, which is relatively long when you consider some of our immune cells. They'll die off faster. And a good reminder is kind of the other organs involved, our liver, we even have our gut involved. But really, if you're able to remember these nutrient pieces here, EPO is your big stimulus. And then the first three and the last two, that's going to help you with the majority of your erythropoietic or your blood cell questions. Okay. One last little funky cell in our myeloid lineage is our megakaryocyte, which we asked about last week on the test. It's the large myeloid cells in the bone marrow. They are going to fragment off into platelets. They can circulate, those platelets then will circulate in the blood and help with blood clot formation and can prevent blood loss. That's their big goal or job. Okay. So the majority of cells are myeloid lineage, right? The other ones, the lymphoid cells, are really our T cells, our T lymphocytes, our B lymphocytes, and innate lymphoid cells, as well as natural killer cells. They also are in this category. So we're going to look a little bit at these specialized cells, and then we'll take a break. So how do we identify, or how do we mature these cells, or how do we tell them apart from each other? They're cell surface proteins, or they're CD molecules, which just represent cluster differentiation. How are they different from one another? And so some important ones to know. Oh, do I have them zoom in? Oh, awkward. Okay, well, we'll start from the bottom then. That'll work. So I don't know. So some important ones that I would want you to know would be CD56. Why? Because it's present on natural killer cells. These are the specific lymphoid cells that it's present on, and they help with adhesion. They're going to help natural killer cells adhere to a location of the body or to their targeted antigen. We have CD28, which we can see present on both of our T cell types, T helpers and T cytotoxic cells, TC cells. These are going to be receptors for co-stimulatory molecules on our antigen presenting cell. Essentially, they're going to help with a part of their APC abilities, their antigen presenting abilities. We then are going to see CD19, 20, and 21. All three of these are only present in B cells. And they're going to signal essentially the growth and development of a B cell from an initial B cell, CD19, a mature B cell, CD20, as well as specifically with Epstein-Barr virus, you can see CD21. That's the only reason why I have that one highlighted, is because it has a specific viral association, Epstein-Barr. We lastly have the most commonly known ones, CD8, which is associated with our T cytotoxic cells or our T killer cells. These bind to MHC class 1 molecules, which we'll see here. So that's the MHC type that they bind to. Versus our CD4 cells are typically associated with our T helper cells. And CD4 cells bind to MHC class 2 molecules. We'll look at this here in another slide with the rule of 8. Okay. So when we differentiate in our body, we form naive B and T cells, meaning naive, they're generic. They're not antigen specific yet. That's what I mean by naive. Once they find an antigen that they have receptors that bind close enough to, they will say, okay, I am now an antigen to, I don't know, pick something. Throw something else. Pollen. I am now an antigen to pollen. And they now will proliferate, and all of their offspring, essentially, all their mitotic offspring are going to have antigen specificity for pollen. And that's how they essentially will become their mature form. Effector cells of any of these carry out specific functions to combat pathogens. Memory cells persist long periods of time in the host. They allow us to respond faster to our next exposure. Our first encounter with an antigen is called our primary response. Our re-encounter is a secondary, you can have a tertiary, et cetera, et cetera. We typically only talk about primary and secondary. So our B cells or B lymphocytes, they mature in the bone marrow. B cells have B cell receptors, BCRs. They essentially are membrane-bound immunoglobulins that can bind to antigens. So this B cell receptor is our immunoglobulin, our IG, that's bound in their membrane prior to being exposed to the antigen that it binds to. It's going to distinguish a mature B cell from other lymphocytes. Each B cell has an antibody that's unique in specificity of seat. Once these bind, there's kind of two special functions that these antigen binding techniques that these B cells can do. One is somatic hypermutation. This makes their binding affinity and avidity increased. It makes them better at binding and locking in and holding on to an antigen. Okay. Also, class switching. It means it can generate multiple types of antibodies. IgM, IgG, IgE, IgA, IgD, I think is even in there. So the ability to class switch is specifically due to our B cells and the B cell receptor. It's their specialized function that they can do. So B lymphocytes, when they are activated, they are the only non-myeloid cell that acts as a professional antigen presenting cell. They can express molecules that are co-stimulatory that can activate T cells. They're actually required. And they will differentiate into effector cells, cells that can plasma cells, which will not have their expression of surface immunoglobulin anymore, and they'll actually secrete immunoglobulin out into the circulation. So you could say that a B lymphocyte who becomes a plasma cell has now lost its ability to bind, but it's essentially gained the ability to secrete antibodies into circulation. Once it's a plasma cell, they do not divide. They will live for years in bone marrow, but the majority will die in about 1 to 2 weeks. This is slightly different than a memory cell. Okay. They do retain some memory, but there is a different pathway for B cells to become memory cells that can live forever, in theory, in our body. Our T lymphocytes or T cells, they're going to mature in the thymus. They also have a unique antigen binding receptor called a TCR, a T cell receptor. So we had BCR, B cell receptor, TCR, T cell receptor. Immunology loves acronyms or abbreviations. Everything's abbreviated. Most of the time it represents what the word actually is. Sometimes it doesn't. but just recognize that that's a very common way of presenting things. Don't let it throw you. It likely means something that's very obvious. T cell receptor, TCR. The TCRs recognize only processed antigens, only peptides. What's a processed antigen? How does an antigen become processed? Phagocytosis, absolutely. So it's only going to recognize an antigen that's essentially presented to itself in a special way. You could think of the T lymphocytes as kind of snooty. They won't just go around and do the scavenging for themselves. They need someone to bring it to them on a platter. That's what our T cells are doing. The antigen that's presented has to be bound to a membrane protein called MHC, our major histocompatibility complex molecules. Our MHC molecules are essentially just sugar proteins, glycoproteins, located on the cell membrane. We saw them on some of our professional antigen presenting cells. MHC class 1 is expressed by all nucleated vertebrate cells. MHC class 2 is expressed only by professional antigen presenting cells. So any cell that has a nucleus can have MHC class 1 in theory. Only cells that are professional APCs can have MHC class 2. The interesting component here is our MHC class 1 binds to or responds to or communicates to our CD8 T cells, our T cytotoxic cells, our T killer cells. This is how we get the rule of 8. Essentially, our TC, our T cytotoxic cells, they're CD8 cells. They respond to MHC class 1. 8 times 1 equals 8. Our T helper cells, they're CD4 cells. They respond to MHC class 2. 4 times 2 equals 8. If you remember your rule of 8s, you'll never forget which MHC responds to which T cell type. Okay, that's consistent throughout the body. It does not change. So our types of T cells, we're almost there. The most histology we'll get to. You have T helper cells, which I've already referenced, TH cells. They have the CD4 membrane glycoprotein. They recognize antigens with MHC type 2 presentation. There are more helper cells than any other type of T cells. This is the majority of T cells in our body. T cytotoxic or T killer cells, TC cells, have our CD8 membrane glycoprotein. They recognize MHC class 1 presented antigens, and they're less populous than helper cells in the body. We need more cells to help and identify things and signal stuff than we do to kill stuff. We have other things that can kill things in the body. Okay. All of these cells essentially are naive, right? I mentioned that at the beginning. They're born, they're generated, not specific for any antigen. When they bind to that MHC, that major histocompatibility protein, that peptide complex, that's when they become activated and mature. They will proliferate, they'll differentiate into their actual adult cytotoxic form. To do this, though, they need a mature CD4 T cell to be able to actually mature and grow up. So we can see here the example is our cytotoxic cell is binding to its MHC class 1. It has to do this in relationship or nearby a CD4 helper cell that's also been activated by that same peptide in its MHC class 2. This is going to then send cytokines over to our CD8 cell to say, okay, I need you to mature now into your cytotoxic form. And that's the signal it's waiting for. This is why we don't have cytotoxic cells just like popping up all over the place in the body, causing everything to become essentially apoptosed or destroyed. It's part of tolerance. A lot of our cells that are intracellularly infected, or a lot of the antigens that are from an intracellular infection, will stimulate a cytotoxic cell response because it's going to bore a hole through that cell membrane and essentially break up the cell and apoptose the cell or kill off the cell. So things that are infected with viruses, because viruses typically infect almost always, actually always, intracellularly, tumor cells or cells of a foreign tissue graft or cells that contain bacteria that infect intracellularly. Those are the things that can activate a cytotoxic T cell. Our THT helper cells will become activated and proliferate similar to CD8. Once they're presented an antigen, they can then mature, become specified, and proliferate into their adult form. There's multiple different types of T cells, T helper cells. I want you to know 4 of them and then 1 that regulates. So that's on the slide. So our Th1 and our Th17. These are our T helper cell type 1 and our T helper cell type 17. Essentially, type 17 secretes interleukin 17, a cytokine. They are going to respond to intracellular pathogens. Okay. Th2 and TFH, F meaning follicular helper cell, are regulating the response to extracellular pathogens. This also would include allergies. This is why we hear Th1 governs infection and Th2 governs allergic response. That's from this here, of where they're essentially responding to the antigen in question. T regulatory cells, these are also T helper cells, but they're going to inhibit the immune system. They contain CD4 and CD25 on their surface. So they are inhibitory. So if there was a question saying what T cell can help quell an autoimmune response or balance the immune system or regulate a Th2 response, Treg is probably your best answer. It's a T helper cell, T cell lymphocyte. It will have a CD4, it'll respond to MHC2. Okay. Any questions on the T helper types? Okay. The last 2 cell types, then we'll break, is our natural killer cells. Natural killer cells are part of our innate immune system, but they come from the lymphoid lineage. They can have a little bit of an adaptive immune function too, but primarily it's innate in nature. They have T cell receptors, which is where they get their adaptive function. And some can even express CD4, although that's rare, but they're not diverse. They, instead of recognizing a specific antigen for pollen or specific antigen for like pine tree pollen, right? They might recognize a generalized protein structure, or generalized fatty acid or amino acid pattern. So they could respond to a more diverse amount of antigens that they're presenting. They don't have to only respond to one type. When they're activated, they are going to release cytotoxic granules that can kill target cells, and they also can release cytokines that can enhance or suppress the immune response. So they have a lot of action. We know the most about them because these were the first studied. So that's why we have the most information about them. Okay. The last one is the innate lymphoid cells. I would care the least about these because they were mainly discussed in the past decade. NPLEX tends to be about 3 years old in its material. And this is just kind of coming into the immunology conversation more and more. So I would learn these the least, but they are a part of the lymphoid lineage. They don't express any antigen-specific receptors. There's 3 different groups broken apart by cytokine secretion, and they help with first line of defense in skin and mucosa. That's really what I would know about these. These are some terms. If you're like, I'm lost, where do I start? Know these terms first, and then you can go from there once you have definitions. Okay. Woo! Deep dive of cells. What questions do you have before we break? There's a lot of stuff. Yeah. Are neutrophils the only granulocytes that's phagocytosed? Eosinophils could. So I would say no, but they have their major action is phagocytosis. So they're the most phagocytosing granulocyte. I wouldn't say only though. Yeah. 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 Th1 could also be an answer. I would say reg is the best one to regulate. Th1 could counterbalance the Th2 because you think of them as like a scale. So if you have a large Th2 activity, typically you're not going to see a large Th1 activity, mainly because your body is upregulating the creation of Th2 immune cells. So I'd say Treg is your best regulator. If they didn't give you that answer and it was asking what could help regulate Th1 or Th2, I'd say Th1 if Treg wasn't present. Does that help? Awesome. Fun stuff. How I'd study, I'd make a little table like we did. Start with an HCS cell, create both lineages, create the categories we talked about, and give them some specific function. One thing per cell type. You can draw that out yourself. you probably know enough to get through this section. Okay, great. Let's break for 10. Stand up, move around, move your body. We'll come back at 11:12, and then we will finish up our last physiology section.